Hello everyone and welcome to Pocket Size Bio of Dr. Casco. In today's video, I want to make a lab activity tutorial in regards to AlphaFold and a little bit of bioinformatics for teachers, right? So I don't see online much resources in terms of using AlphaFold and a lab activity for high schoolers or even undergrad. So I came up with a lab activity that I did with my students, and they seem to really enjoy it. So I'm doing a kind of a walkthrough of that lab activity. I've tweaked some stuff for this little document that you see here for teachers, and then you can change and modify it for your classroom. I hope this is helpful for you. It was a very positive experience for my students and myself, so I wanted to share this with other educators. So let's get to it. More specifically, my lab activity is based on comparative analysis between AlphaFold predicted structure to an experimentally deduced structure using molecular docking. And I gave this class during the macromolecule section of Unit 1 in the AP curriculum, right? So right after I talked about proteins, and just to give you a sense, right after I talked about proteins and their structure and the amino acids and whatnot, I gave this lab. More specifically, I gave it right after I discussed in detail the primary, secondary, tertiary structures and quaternary structures, so the students could get a sense of what the alpha helices and the beta pleated sheets look like, and just how modeling looks like in general. So they'll be engaged in that in this activity. And then I gave a little bit about kind of context on how scientists are able to calculate and create 3D models, right? Experimentally validated models using X-ray diffraction. And then I got into AlphaFold, right? This is a relatively recent breakthrough, especially with AlphaFold 3. So it's a very nice thing to show to the students that's kind of frontier science. And if you have followed up with AlphaFold, this is an AI-based tool that can predict protein folding from DeepMind of Google. And they even won a Nobel Prize. So I think the students were very interested in that. So going into a little bit more detail into AlphaFold, just to put into perspective for you, so you have this problem, this protein folding problem, where it's quite difficult to determine the 3D structure of a protein, of a polypeptide, from just the amino acid sequence, right? And there's several folding ways in which a polypeptide could go, and being able to predict how it could fold into is one of the the biggest challenges of the from 50 years. So AlphaFold does exactly that, right? It can predict the 3D structure of an amino acid sequence and give you an output, right? And why is this particularly important? Many reasons, but one that I focus on with my students is for drug discovery, knowing some potential functions of the protein, right? Because of that idea of the lock and key, and finding active sites. And one of the research that was implemented using AlphaFold that was pretty big was EGFR in cancer, right? So researchers they studied a predicted protein from EGFR that I'm going to talk about in a little bit, and they were able to find a drug for a type of lung cancer. Initially using AlphaFold. Of course, they validated that 3D structure, and that's what we're going to analyze today. So the lab activity in itself is comparing a a AlphaFold predicted structure versus the actual experimentally deduced structure that these scientists use. All right, so going on into the actual activity here. Again, as I mentioned before, with the topic, how does an AlphaFold predictive protein structure compare to an experimentally deduced structure when docking with the same ligand? So we're gonna put into the test the ligand and also how that ligand or that drug fits into the experimentally deduced structure and AlphaFold structure. So how well can AlphaFold actually make the structure too? Okay, here's some learning objectives, explaining how protein structure determines function, basically the active sites, retrieving and analyzing 3D models, right, the PDB format we're going to take a look at, and several tools other than molecular docking that we need. And then, we're going to compare by the binding site. So this is a metric that can tell you how well the drug will bind to the active site or that particular site of the protein. Okay. As I mentioned before, epidermal growth factor receptor, so EGFR, is that protein of interest, right? It's a surface receptor that comes and collects and binds to a signal, which is an epidermal growth factor. And that relays a cascade of signals all the way to the DNA, ultimately to regulate cell growth, division, and survival, right? So in many cancers, though, this EGFR becomes overly expressed. So it's mutated and it's always active, okay? Of course, that leads to uncontrolled cell division and tumor formation. And it's because of that, it's a good target for drug discovery. So one of the drugs that this research found was gefitinib. It's a small molecule that binds to the ATP binding pocket, right, or that binding site. It's and it's on the receptor kinases domain, and it blocks the signaling activity. So that's what we're going to assess. I've provided some additional information for the students in terms of the protein target. In this case, our protein target is EGFR, and our ligand or our drug is gefitinib. Okay, and this is what we're gonna work with. Okay, so let's get into the actual methodology. I think this is what's important. I've placed here several links, several websites that we're going to use. By the way, everything could be done on a tablet, on a laptop, because it's web-based. So all your data and files are just submitted into the web format. So the students just need to know how to navigate their folders and things like that. Renaming is important. And such there. I have seen successful runs on the iPad. I have seen also on the PC. Some students have had difficulties on the Mac for some reason for the website to run, but the majority were able to get it done successfully. Okay, so I've split this into parts. The first part is retrieving and preparing the PDB protein structure files. So these are 3D structures of files that are in the format called PDB. There's some other formats that we need to turn into, convert, that we're going to take a look at. So for this first part, we need to get a file from AlphaFold, which is the 3D predicted, and the experimentally validated protein. Okay, so the first thing we're going to do is get the amino acid sequence, okay, of this EGFR protein. So we go to PDB, Protein Data Bank. All right, and I'm going to be working here now. Everything I'm doing, that way you can continuously see my steps, my procedures. And we're going to get that this protein ID. Now this is the same ID as the experimentally validated protein. Okay, crystal structure of the mutated EGFR kinase domain in complex with gefitinib, and I'm going to get into that in a little bit. But right now, I just want to collect the amino acid sequence. Okay, it's this one here, 3UG2. That's the ID of this submission from the researchers. All right, so you want to go to display files, okay? And you want to get your FASTA sequence. So this is the amino acid sequence of the EGFR for Homo sapiens, the chain A more specifically. And I asked the students to just paste, their sequence here. Okay. And I usually put this in a little bit a smaller sequence there so they can see it more neatly, right? So you have your sequence here. Then what you're going to do, you just want to copy the sequence only without the identifier, the sequence identifier. Okay. This particular format that you see here is called FASTA format, and it's just a standard way to write amino acid or DNA sequencing followed by the name identifier. Okay. So you want to copy your sequence, and now we're going to go to the AlphaFold server. So we go to the next link. I'm going to move this here. So this is your AlphaFold server. You can log in with your Google account, and you can see you can have a history of all your runs. You notice here I've done it several times. So you're going to go to add entity, and since we're working with amino acids, we're going to be putting protein, and then you just input your amino acid sequence. Okay. You might have some errors where you have spaces there, and you can see there's a little red line there. You can just fix that just by putting a space, and it should be okay. So you put continue and preview job. It's always good to rename your jobs, right? So you don't get confused there. So I have EGFR, and I'm going to put this as C since I've done this run several times. Okay, so confirm and submit job. All right, so this might take a little bit of time, and you can see it running here. Okay, so since I've done this several times, I'm just gonna click on it, and you'll get this particular output. So just letting it load it for a bit. So when it finished the run, you can see the 3D structure of your EGFR protein made by AlphaFold. And some things about this. Now, this lab activity doesn't go into detail with what we're seeing here in AlphaFold per se, but just to give you a sense, the coloration indicates the confidence level of the structure that you're seeing here. So blue areas have a confidence metric. This metric is called PLDDT, and if it's blue, it means that it has a confidence of 90 and up and above. So that's pretty good. Ideally, we always want to have as much blue as possible, right? And then it'll go lower and lower with the different colors. You will have some very low coloration, and it's normal. There's some protein areas that are disordered regions, that are flexible regions, that it's a little bit more difficult for these predictive websites to calculate. But as long as you have these main domains, that's what's important. Right here on the side, this is a PAE score. And this is a metric that kind of shows the domains and how these domains are kind of built in the 3D space relative to other domains. For now, what you need to know is that the more green you have in this diagram, the better, right? Now, they do get more specific depending on the different types of protein, where you can see just very specific squares along on this matrix. But for now, that's all really you need to know. If you would like to take a screenshot of that, that's fine. But one thing that you do need to kind of record, or that the students should record, is the PTM score. This is an overall score that tells you how good the build was for this particular protein. I have seen studies using 0.5, and it has been pretty good. This is a score of 0.9, which is really, really good, right? So in this case, you can ask the students to just write 0.91 as their value for that AlphaFold. Okay, so I asked my students even to take a screenshot of their output there. Okay, and you can just dump it inside their data file. Okay, showing them that they got an actual structure and some good metrics there. Okay, so what you're gonna do is download, go here to download the actual 3D file, which is in a file we need to convert. So, okay, you're gonna get a zip file. So save it to somewhere where you can work with pretty well. Let me just rename this. We're gonna name it C. Okay. Good. So we're gonna open this zip file, and among this zip file, you want to extract. Let me just click on that. There you go. You want to extract one particular file. So the ones that say model 0. Okay. So here's where I'm talking about model 0. So these are the 3D models that AlphaFold makes, and it makes several. The model 0 is essentially the best one, but it has several forms that it created. For the sake of time, I only use model 0, but students can explore all these different models. You can explore the different models too. And so you want to extract that. Okay, so I've extracted that one. So now we have model 0 here. And you should rename these models, these names, into AlphaFold EGFR, right? So AlphaFold EGFR. So I already have it here. Okay. For Mac users, I noticed that it was a little bit difficult for some students. You want to make sure that you have .cif at the end of the naming. Okay, so make sure you have .cif at the end of the naming. Okay, don't put a period there. Just at the end, you want .cif. Okay, so let's continue on. We need to convert this CIF file into the standard PDB 3D structure file. And the way you do that is that you go into this website. Okay, this is a simple converter from CIF to PDB. And you choose your file. Since I have it here, okay, and you can see that it's in CIF, this crystal information file. You click it, and let's put download PDB. Okay, so you want to name it in the same, you want to have it in, the same area that you're working in. In this case, we have PDB. You have now the PDB file that's going to be saved. All right, so now we have our PDB version of this particular file. So now we have our AlphaFold file. We don't need this website anymore. You can close that. And you can also close the AlphaFold server for now. Okay. Now we need the experimentally validated 3D file. Okay, we're done with AlphaFold. So we're in step 4, obtaining the experimentally validated EGFR PDB file. So you want to go back here into the protein data bank, and you go to download files. So you have this legacy PDB format. Okay, so it's going to download. It's already in PDB. Okay, so for this, you want to rename. Let's name this one to experiment exp EGFR. And I want to say with, and I'm going to explain this in a second, with gef. Okay, with gef. So that means gefitinib. Experimentally validated EGFR with gefitinib complex. Okay, and I'm going to explain what this is. So this file that we just downloaded is this 3D structure. And I'm going to go to Explore in 3D. Okay. And this is a model viewer. Okay. So if you notice, this is the EGFR that the researchers actually did the X-ray crystallography, and I'm going to explain that. Okay. And they mention it's in complex with gefitinib. Now, what that means is that in the 3D structure, we have the drug also, which you can find here. Where is it? There it is. Okay. You can just click on ligand. Okay. If you notice, the drug is already here. Okay, and it's highlighted in pink right there. Okay. So in this file, we actually have to remove this ligand before we can test it against AlphaFold. Okay. All right. So the way that we do that, let me just, okay. Okay. Oops. Let me just fix that. I needed to add an additional step. Yeah. Oh, sorry. I do have it here. Okay, so we got our protein structure there. So if you see the drug is in the 3D structure, we need to remove that. And there's some online websites that can do that too. So we're going to go to PDB Tools website. So let's go back up here, PDB Tools website. So let's click on that. Okay. And before I continue this, I just want to show you some information in terms of the experimentally validated protein. So here, this is some information that you can use that the students can extract. Okay. The method of collecting the 3D structure is X-ray diffraction. The resolution is 2.50 angstroms. Okay. So scientists use generally angstroms that are 3 and below. The lower, the better. Okay, but I've seen studies that have successfully published with 2.5, and so forth. And if you navigate PDB website, you're going to see many of them are around that range. So the quality of this protein is really good. The students can take that information and they could put it here if they would like, or if you would like them to collect that information. Okay. Okay. So again, now we need to kind of clean up the protein. So the way that we do that is that we go to pdbtools.web and we're going to upload our file. So here, if you remember, we got experimental, right, EGFR with gefitinib. So let's click on that. Let's upload. Okay, just takes a little bit of time there. Okay, one structure has been successfully uploaded. And you want to go to that main category now. So this is number 6, main category. And you want to drop down, click the drop down button, and go to PDB delete het atom. Okay. So what this is going to do is going to remove all of the ligands and hydrogens and water solvent that are floating around in that 3D structure. So I want to show you. There's a little bit of impurities within that within the structure there. So if you notice, there's a bunch of water molecules that are floating around in red, and there's some other ligands in additional to gefitinib. Okay, so we want to clean that up. So we go to success, everything's good. We click on run. Let me check here, PDB code. So we got our upload, parse, main, good. Okay, pre-processing, choose file, upload pipeline. Oh no, just a second here. Okay, so you select, sorry about that, I forgot to put the plus here. So you put pdb.del het atom, and then you put plus. So now we have a little pipeline that we're going to do. And then you can play, put play, run. And it's going to start running. Okay, so it finished successfully. It goes very quickly here. And now we can take a look at our protein molecule. And um, there's no water molecules floating around, and there's no ligand floating around. Okay, so it's nice and clean. Good. So you can download this output number 1. Of course, we can rename it. You should rename it. And now we're going to say without gefitinib. Great. So now you have cleaned your protein. Okay. Continuing on. So now we're going to prepare the ligand. So this is a very simple step. You just go to PubChem. PubChem is a database of many, many molecules and chemicals and drugs that scientists have deposited. Okay, so let's search for gefitinib, and you're going to look for a formula, a SMILES formula. If you see here, okay, this formula, it's kind of code, a standard code that scientists use in informatics. Okay, and you can just paste like that, or students can paste like that. That equates to the formula here, gefitinib. And if you click on gefitinib, if you want the students to take a screenshot to be familiar with the structure, it looks something like this. Okay, you can change the different type of format. Very nice wireframe. Okay, that way they can get a sense of what they want to look for in the 3D model. Okay, so we can do that. So it's a 3-ringed structure essentially. Okay, so you can copy the file there, and then we can paste your molecule, gefitinib. Okay, that's it. This is all we need, the SMILES formula. Now we're going to go to pRank Web, and this is a website that predicts where the active sites are. So I'm going to scoot this over here. Okay, there's a couple of things that you can write down. We're almost done. So you want to go to custom structure, and you want to choose your file. Okay, so I will choose the PDB. Remember, we're working with PDB of AlphaFold. Okay, you leave all these parameters in default. Okay, leave all the parameters, take a screenshot. Let's take a look. Okay, so it's going to start running. So the information you see here is just code that it's running. Tell the students to wait for it. Okay, I'm going to open it again so I can run the experimental. Okay, so let me just upload now our cleaned experimental without gefitinib and submit. Okay, so now we have pRank Web running. You should have something like this. Very, very nice. So what we're seeing here is, I'm going to make this bigger just for you to see what I'm looking at. So this is your AlphaFold 3D structure. You want to have the actual protein structure in cartoon form. Okay. And then the, let me just move this here. The binding site, I like to have it as surface residues. So this website gives you a predicted determination of where the ligand is most likely to bond. In this case, it's in this red area here. They're referred to as binding sites or pockets. In this case, we're calling them pockets. And things that the students can collect in terms of data is the 3D structure, okay? And we can put that for binding pockets here. So you want to take a screenshot of the area, and let's put AlphaFold EGFR. Okay, put paste. You have your model there. Okay, and you also want to collect the score, right? The pocket score. So you can put this at the bottom here. I don't have a table for that, but let's run the content, right? So pocket. So in this case, pocket score. So let's say 22.00. All right, so this gives you a relative value of how good this score, this pocket is. I have seen studies that use a score of 4 or 5, and they've gotten some good results. So 22 is definitely a good thing. Now you want to collect some additional things. The pocket center. So here is where your ligand is going to, the algorithm is going to interact the binding site with your ligand, with your drug at this particular coordinate. Okay. So this is also something that you can write down. Since I'm working with AlphaFold, you want to write these coordinates here. Okay. And then you'll do the same thing with the experimentally validated one. Okay, so the experimentally validated one got 10.53, which is still okay. It's relatively high, but the pocket center is a little bit different. So we can copy that and let's put the center there. Okay. And again, same thing. You want to change the protein to cartoon and change the binding area to this little surface residue site. Quite similar. Not identical, but quite similar. And then you can post it here, right? So now we have EXP EGFR, and this is your protein. Ideally, the students can have the same shapes there. Okay, and then, sorry, Beth. Okay, so now we're done with the binding, finding the binding pockets. So we've predicted that we do have a good binding pocket, and technically the drug should be there, all right, when we do the molecular docking. So the molecular docking, this is the last part. Okay, now I'm going to open 2 and move them over here. Whoops. Okay, Swiss doc and Swiss doc. For this last part, you want to choose AutoDoc Vena. Okay, AutoDoc Vena, and we can plug in the same Smiles code, okay, because we're using the same drug. Where am I? Here we go. Okay, so you want to put that. You want to prepare your ligand. Okay, and then you submit a target. So in this case, we're using AlphaFold, the protein data bank here. Okay, then you want to prepare the target. Okay, it's going to get a check if everything's okay. And you can see now, you can see your protein, and then there's a box here. This box is the space in which the algorithm is going to look for the binding site and the drug interaction. Okay, and you can manipulate this box and move it around. So with AlphaFold, we had these particular coordinates. So you want to put the center, the box center or the pocket center, you want to put it in. So we had negative 7.47, right? Enter. Oops. No, why didn't that work? Negative 7.47 or 48. Okay, if you notice the box changed a little bit. Let's put the next one, 0.04. Okay, and then the last one there, 1.94. Oh, sorry. Negative 1.189. Enter. So this should be our box area. Now you could make the box a little bit bigger by increasing the box size, but there is a possibility that the algorithm in the website could give you an error that the box is too big. So you don't want the box to be too big there. Okay, but this seems good enough. Aside from that, going back to the steps, okay, so we prepare the protein, and you want to put the exhaustivity is the number of runs that the algorithm will go to see how well the binding was. I usually put it at 8. 8 is kind of the standard. And then you put check parameters. So it's going to verify if everything's okay in terms of the conditions, and then it's going to start, it will enable you to start the docking. If you get an error, then you should lower the box size. Don't do anything with the box center. Okay, so it checked, and you can do the docking. Click on the docking. All right, so then it's going to load this kind of progress bar. We're going to continue with the experimental without gefitinib. All right. If you notice, it's already here. Very nice. Okay, let's prepare the box. Let's prepare the target, and let's put the box coordinates here. So in this case, we have 1.94. Enter. We have 50.81. Enter. Now, you cannot use the same box coordinates for both of them because they're just different files, and the different they're in a different space when they were created in the software. So every protein that you use will have a different space. Sorry about that. Okay, this makes the box a little bit bigger. Okay, exhaustivity to 8, and check parameters. Okay, it's still running there. It should be okay. If there's an error, it's gonna give you a prompt that is you're exceeding the size. Okay, so we're gonna start docking. So now these two are running. I will pause the video now until it finishes. Okay, so the AlphaFold run finished already. So you can see the data information here, AlphaFold EGFR. Again, you want to have identical, box center and box size. You can write this down. We used 8 for AlphaFold and experimental, and you want to have your coordinates for box size here. So you can copy and paste that there. Okay. So this one in blue is your protein by itself. So don't worry about that. Now, to see your actual protein, you want to click on number 1, and your interaction is now found here. So this is what the students need to go for. Now, in this ribbon form, it's a little bit difficult to see, like, the beauty of these computations. So you want to go to surface protein, show protein surface, and your docking is right here. Okay, you can even see how it fits amongst this groove here. Sorry about that. Okay, so this is your result here. Now you can take off this protein surface, and you can click on these additional interactions. Okay, but this is a little bit beyond the scope for high school, but maybe for university, if you want to see these types of interactions, you can even see some little interactions occurring there. You can go deep into seeing the amino acid residues, but that's a little bit beyond the scope. So you can take a picture of this particular interaction, okay, and put AlphaFold here. Okay, and then the last thing you want to write down is your binding energy. This is the most important factor here. That this is what shows how well the docking is. I've seen binding energies of -3 be fairly well. So you want to record this. This gives a degree of stability, right? That you have this binding area occurring fairly well. Okay. And just to look at the experimental one, again, you have your binding, your box here. We can paste it here, and let's take a look at our auto affinity. Okay, so pretty close here. let's take a look. Oh, so this molecular docking seemed to be even more to fit at that active site, but they seem to be in the same position, and they seem to be also in that same affinity. Okay, so AlphaFold did a pretty good job in kind of predicting this whole molecular docking, and that's the whole purpose of this experiment, right? We're kind of challenging, one, how good the ligand is, two, how good AlphaFold, the AlphaFold, moreover, is at predicting the structure, and then how good the whole ligand in itself is at binding to this particular thing. Okay, and we can put our data here, negative 6.9, 3, 1, 3, 1. Okay, so there's some post-lab questions, analysis questions that I've given my students that you can use with this Google Doc. But in terms of the actual bioinformatics procedure, that's essentially it. So I hope this video was useful for you. The whole lab activity was quite insightful for the students. So thank you for watching. I do want to mention one last thing for P rank that I should have collected. I didn't collect this with my students, but it would be good to collect, is the probability, right? The probability, so this is of course, probability on how the likelihood in which the ligand will bind to that, to the binding site. So 85, 86% is really high, okay? And the same thing with here. So this one, you have this other protein here, okay? Probability 60% is quite interesting, being the experimental there, right? But still, that's a pretty good value, okay, for the efficacy of the protein and the drug interaction. Okay, that's something you can add in terms of the data table, but of course, everyone's welcome to do whatever they would like in terms of what they want their students to collect, right? Okay. So thank you very much for watching. I hope it was helpful. If you are interested, I can make different types of bioinformatic lab activities. Okay. Thank you for your time.